The human gut operates as a massive shared workspace where every single microbe passing through is strictly monitored. When we consume premium probiotics, these friendly bacteria enter this environment, searching for a stable surface where they can attach and begin to grow. However, if the host environment lacks sufficient vitamin D, the local immune system identifies these new arrivals as a potential threat. Without the proper biological signal to stand down, the immune system prevents the microbes from settling. They remain in the open space for only a few hours before they are physically flushed out of the body. This explains why increasing the dose of a probiotic supplement often fails to change gut health. The bacteria cannot colonize an environment that hasn't been prepared to receive them. When vitamin D3 reaches the gut lining, it acts as a primary signaling molecule that dictates how the digestive tract interacts with microbes. On the gut cell's surface are vitamin D receptors, VDR. Vitamin D attaches to them, triggering a chemical response. This signals the mucosal immune system to remain calm. With the immune response neutralized, incoming probiotic partners safely approach and attach to the lining. This molecular interaction changes the gut from a defensive barrier into a cooperative habitat where beneficial bacteria can finally survive. Once these microbial partners are established, vitamin D coordinates the physical reinforcement of the gut wall. The cells begin producing tight junction proteins. These act like microscopic staples that pull the cell walls together, making the entire lining watertight. At the same time, the cells secrete mucins, forming a thick, protective fluid layer that coats the newly sealed barrier. Deep within the lining, Paneth cells begin releasing alpha-defensins. These are smart proteins that selectively target and eliminate harmful bacteria while leaving the friendly probiotic colonies unharmed. In this state, vitamin D is managing the construction of a physical and chemical fortress that protects both the host and its microbial partners. As the probiotic colonies grow, they perform their primary task, fermenting the dietary fiber that passes through the gut. This fermentation process produces a short-chain fatty acid called butyrate-sustaining loop. The vitamin allows the bacteria to settle, and the bacteria help the body create more sites to receive the vitamin. When vitamin D levels are too low, this cooperation breaks down. The microscopic staples holding the cells together begin to fail, leaving open gaps in the gut wall. Toxic fragments from the walls of bad bacteria, known as LPS, leak through these gaps and enter the bloodstream. These fragments bind to immune sensors in the blood, keeping the body's defense system in a state of constant low-grade alarm. This constant immune activity creates biological noise that interferes with cellular communication. Eventually, cells stop responding to the signals that tell them to absorb sugar from the blood. This signaling failure is the mechanism behind insulin resistance, showing how a lack of vitamin D in the gut can lead to systemic metabolic issues. When vitamin D levels are optimal, the gut wall is sealed, the immune system is balanced, and beneficial microbes can thrive. True digestive health depends on this readiness. Probiotics can only do their work when the host has provided the necessary environment for them to live. By ensuring the body is prepared, we allow these trillions of microscopic partners to settle in and work in synchronization with our own biology.